in terms of some of the terminology, what I've done is this. We kind of identify a major level from that standpoint. And then from that end, we're going to identify the division, some key structures at that point. Will help define these for you and kind of go there. Malcolm, before I continue, are there any immediate questions I need to address or like to address?
We have a question here from a student that what book or study guide would you recommend to study for the MRI ARRT exam?
Well, now, is that in general or is that just for sexual anatomy? I guess I might be better suited to address that. That one's a hard one. I think there's a couple that stand out. One is by Kelly Peterson, Lori Kelly and Connie Peterson actually taught up at Boise State. They do a good job in terms of identifying key landmarks of sexual anatomy of the body. And that they cover a full gamut of different modalities. There's another one by Michael Madsen out of Fort Hays, Kansas. He did a pretty good job with his book. But I'll be honest with you, if you really want to provide the best resource for learning sexual anatomy, is to take information I'm hoping that we give you today and look at actual images. What you need to learn is contiguous slices. You need to start from the top, work from the bottom, and piece together these different landmarks. The only issue I've had with textbooks that I've tried to use is that they jump around quite a bit. They'll show you one, for example, T1 level of the upper cerebrum, then they'll jump down to the cerebellum, they'll show portions of the brainstem, then get into vascular structures. And the way they present contiguous slices oftentimes is haphazard. That's really where I get frustrated with most textbooks. So here's what I'm going to recommend. I'm hoping what we give you today will give you a reference point to work from. And then I would get your own images and piece together a collection of images that help you identify what these key landmarks are. So basically, there's a couple good books out there, but I think until they really produce a resource that provides contiguous slices from top to bottom, it's hard for me to adopt a book. Other questions at this point?
That's good. We've just had some comments that people are at the edge of their seats, and it's really fascinating. So I think all is going well.
Good Lord. Okay, let's see if I can live up to that. All right, here's the mid-sagittal slice. If you ever get lost in terms of identifying a key structure, I tell my students this always. When we look at a given structure, for example, if I'm looking at the anterior portion of the corpus callosum on a transverse slice, I can't identify it. I always have them go back to the mid-sagittal slice. It gives you a roadmap. It gives me every direction in terms of where I'm moving from top to bottom, anterior to posterior. It gives me lay of the land. Right back over here, for example, is the tentorium cerebelli. Above it is a venous sinus called the straight sinus. When I'm looking at then the transverse, this will give me a roadmap. I get lost, I go back to that, okay? With the mid-sagittal slice, here are some key features, key landmarks I want you to be able to identify. Number one is one we've already talked about, which is the connector. This is the corpus callosum. And once again, I have the anterior inferior limb called the rostrum, anterior genu, mid portion body, and then posterior portion is called the splenium. These two ends here will stand out on a transverse slice. The anterior limb referred to as the genu. Again, here's the splenium. We'll see that the lateral ventricles are found in and around the corpus callosum. And we'll talk about the ventricular system rather briefly. We'll kind of go through that. You have thalamus over here. That's what this large bulbous gray matter structure is. There are paired structures. We're going to find that they form, I mentioned it before, the lateral walls of the third ventricle. It nicely lays out the brain stem. For those getting ready for your registry, please keep in mind there are three components of the brain stem, namely, get my pointer back here, midbrain, pons, medulla. So this area basically, or this image, will kind of lay that out for you. Here, behind the midbrain, go back a step, cerebral peduncles right over here, Mickey's ears, seen on end, Mickey's head, sorry for using the Mickey Mouse metaphor. Here's the mouth of Mickey. We'll talk briefly about that when we get into the ventricular system. And what this is basically is the passageway between the third ventricle, which we can't quite see, it's being obscured by the thalamus, leading into this somewhat of a diamond-shaped structure over here, which is the fourth ventricle. That little line right over here is going to be the cerebral aqueduct. Here's a point I do want to make now, in case I forget to mention it down the road. As you're getting ready for the national boards, keep in mind that the ARRT, I can't speak to SDMS, use Gray's Anatomy as a reference point. So one of the things that as an author we've had to do is make sure that any terminology is updated so that what we give our students, the reader, is correct. This is a prime example right over here. It used to be called the aqueduct of Sylvius. In new terminology, it's called the cerebral aqueduct. Back over here, we used to call this the quadrigeminal cistern. Now it's called superior. Here we used to call this the quadrigeminal plate. Now we call it the tectum. So I'm going to try to always give you the most current terminology. As you prepare for your national boards, make sure that you have the most current terminology learned from that standpoint. Continuing right over here, we're going to get into cisterns. We don't have a lot of time to get into cisterns. Basically, we know that the CSF within the brain and around the spinal cord is contained within a region beneath the second layer of the meninges called the arachnoid mater, that space being the subarachnoid space. When you get enlarged areas in and around the brain that aren't ventricles, but still expansive areas, we give those the term cisterns, like a catchment for water. What you see over here, which might appear to be air-filled, in reality is a cistern. We'll talk about those briefly. So you have cisterns, you're going to have ventricles. Here is sphenoid sinus, which is air-filled. Again, we cover those for the most part. So again, this is going to, clivus right over here, and that's the lower portion of sphenoid bone that forms eventually the anterior portion of the foramen magnum. So always go back to this from that standpoint. Hopefully the terminology will help you. So I can get this to advance. Other structures, we talked some already about these, so I'm not going to spend a lot of time with that. I do want to distinguish between thalamus and hypothalamus. Here's the thalamus, and these again are large gray matter structures that are paired and basically are interpretation centers, relay centers from one portion to the brain to the other. Right over here is the hypothalamus. The hypothalamus, smaller in scope, anterior, inferior to the thalamus, and it's going to help regulate things like homeostasis, which controls pH, body temperature, pulse, things like that. So you have the hypothalamus over here. You're going to have the thalamus, larger, more dominant from that standpoint. Pineal gland, which is going to be found basically adjacent to the roof of the third ventricle. We can't quite see it on this one. We'll show you another example of it. Pineal means pine cone. As an individual gets older, they sometimes calcify. They produces melatonin, which helps promote sleep, things like that. The tentorium cerebelli is right over here. From that standpoint, you'll see it someone has a white density or somewhat of a brighter density through here. Above it is one of those, not cisterns, rather venous sinuses that are going to emerge away from the area of the falx cerebri. This is going to be called the straight sinus and eventually will join up with the superior sagittal sinus. The first upper cerebrum structures we want to talk about once again is going to be the mid-sagittal portion or the longitudinal fissure right through here. It's the dividing point between the largest hemispheres of the cerebrum. It's going to be directly midline from that standpoint. Once again, within it, you're going to find that it's either the fissure that we identified for you or a portion of the falx cerebri, or, is what we see back over here posterior, it could represent a portion of the superior sagittal sinus. So this area can encompass 3 areas from that standpoint. Over here, we talked about that burst of white, here and here. These perhaps are the most dominant portion of the upper cerebrum. It's called the centrum semiovale. These are large collections of white matter tracts. You can see how massive they are over here. Again, I already mentioned briefly before, these impulses, both sensory and motor, are going to be carried from the spinal cord area up into the brainstem. We'll talk about structures called pyramids. Pyramids is where those nerve fibers will actually cross, which basically, you've heard it before, the right side of the brain controls the left side of the body and vice versa. The left portion of the cerebrum will control the opposite side of the body. These basically are going to be these large collections of white matter tracts. And eventually, much like a fountain, they're going to come up, they're going to eventually divert, and their impulse go to different portions of this layer right through here, which is the cerebral cortex. Cerebral cortex, really interesting area. It's a layer of gray matter structure, probably no thicker than 3 to 4 millimeters in thickness, yet it's really important in terms of higher order thinking, problem solving, rational thought. And basically, it's going to help us in terms of being able to take all these impulses from the body and begin the process of problem solving from that standpoint. But from the upper cerebrum area, centrum semiovale are really the most dominant of these structures. As we work down toward the cerebrum, working our way down to the second division, we see right off the bat the lateral ventricles. The lateral ventricles are large landmarks, and they're going to give rise or help us identify other structures in and around them. First of all, we see that you have an anterior portion of white matter tracts that's going to connect both hemispheres of the cerebrum called the genu. There's another band of white matter tracts posterior called the splenium, again, components of the corpus callosum. The lateral ventricle, we'll talk about this area right in a second. This might be a part of the body. The lateral ventricles are interesting. Historically, a little background, we go from lateral ventricles to the third and fourth. So the question is asked, why not first, second, third, fourth? The reason is, as you read on the history of it, early anatomists would go through, these we're talking back in the 1600s, they would actually open up the brain on cadavers and then look at it, and they saw these cavities and they called them these cavities called ventricles, but they can never settle on which was first and which was second. Some anatomists felt the one on the patient's left or right, rather in this case, was one, the other one was two, and then they would argue about that. So early anatomists decided just to call them lateral ventricles. They're very dominant. You're also going to pick up the first of gray matter structures. We'll talk about these, are basal ganglia, right over here. So when you look at the landmark of the lateral ventricles, you have them stand out. Once you see them, you pick up the genu, you pick up splenium, and then we're going to talk about these here in a second, called the caudate nucleus from that standpoint. These stand out as key landmarks from that from the standpoint of this portion of the cerebrum. You still see an element of the longitudinal fissure. You see it over here. And then again, you see that little triangular shaped defect right here is a collection of blood referred to as a superior sagittal sinus. Again, all that terminology is for you on the left column. Ultrasound, you're able to get a midline neonate. Here's the midline, and then the ventricles would be on either side. We won't spend much time on that. We'll kind of move on. Continuing down the cerebrum, you get now better definition of the lateral ventricles from that standpoint. And what you see right here is a line, a lamina, a membrane that separates both of the lateral ventricles, called the septum pellucidum. So the septum pellucidum is a really important landmark right through here. You see it kind of stand out. It's a midline structure, divides both of the lateral ventricles over here. Once again, structures we talked about, genu, splenium, and then you pick up right over here, directly adjacent to the lateral ventricles, the first of the basal ganglia, called the caudate nucleus. The caudate nucleus is always going to be associated with the landmark of the lateral ventricles. Going from anterior to posterior, get my little cursor back, you have right over here three divisions of the caudate. The one that's going to be the most dominant is along the anterior portion or anterior horns of the lateral ventricles, called the head, mid portion, body, posterior portion, tail. So you have the three divisions of the caudate nucleus. You have genu, you have splenium, you have over here septum pellucidum, and then the two lateral ventricles. That basically kind of stands out as the key landmarks in that portion of the cerebrum. Continuing down, and I'm taking these in very incremental slices, so we're not really seeing a lot of change or transition, so I don't miss any of the key anatomy. We see, again, the same structures at what we saw earlier, but I want to point out these little white areas right through here. We'll see a much more dominant as we work our way down to the third and ventricle and other portions of the anterior posterior horns of the laterals. These are called choroid plexus. They're specialized capillary beds found in and around all of the lateral or the ventricular system. Basically, what they're responsible for is through a process of both osmosis and what we call active transport. It's going to take the blood plasma. It's going to extract certain electrolytes, plasma, of course, certain elements, certain minerals, and it's going to produce by the process of this active transport, CSF. These structures here are going to be producing cerebrospinal fluid 24 hours a day, 7 days a week, and it's always in a regulated, balanced manner that they're producing the CSF, and it's going to basically flow then through the ventricular system, the CSF, through the different openings, through the different cisterns, in and around the brain, and eventually down into the cord. When the CSF is expended, there's little finger-like projections called arachnoid villi granulations that then draw the expended CSF back into the venous circulation. We won't spend any time on that, but these little dots in here are always going to be referred to as choroid plexus. You'll see them within the lateral ventricles. Once again, taking you around the horn, genu, splenium, septum pellucidum, right through here, lateral ventricles, the spaghetti-like structures in here, choroid plexus. Once again, any gray matter structure adjacent to the lateral ventricles would be caudate nucleus, specifically head, body, tail. The falx cerebri basically is going to be this little area white in this midline portion of the cerebrum. Again, it could be a portion of the falx, it could be a portion of the fissure itself. In some rare cases at this point, you might be able to pick up a little bit of a superior sagittal sinus. We're getting into the basal ganglia area, and this area is really rich in terms of a lot of important landmarks as you work your way down. Notice this from a transition perspective. We've gotten away from the body of the lateral ventricles when we have our two extensions of horns. We have anterior and posterior horns right through here. We also get an early sign of the third ventricle right here. We'll look at that in a little bit more detail. Structures we've already identified. Once again, we'll reinforce that. Genu, splenium, back over here. The spaghetti-like structures in here, choroid plexus. Here, along the anterior horns, would be the head of the caudate nucleus. Over here is going to be a second group of basal ganglia. We'll see that they're paired. In fact, these structures are typically paired. And this is referred to as a lentiform nucleus right over here. The lentiform nucleus was described by early anatomists as being a shape of a lens, thus the term. But what they found out later, and we can't quite see it on my slides, perhaps on a higher resolution MR image, you'll see a small membrane or lamina that tells us the lentiform nucleus is made up of two components. The medial one is called globus pallidus, and the one that's lateral is referred to as the putamen. What basically the globus pallidus does, and most of these will have an impact in terms of motor function to a certain degree of movement of large and small muscle groups. The globus pallidus helps us regulate motor function in terms of muscle groups. The putamen is kind of interesting. It helps us coordinate our movement, our ability to move in a certain pattern, rhythm, or gait in a certain degree is controlled by the putamen. One prime example is that when we walk, sorry, I know I'm kind of getting animated here, that when you move your legs, your arms are going to follow in a certain sequence. That is done primarily in terms of balance and maintaining a certain stride and gait. That's being controlled to a certain degree by the putamen. So here again, we have two of these dominant basal ganglia, caudate nucleus, lentiform nucleus, medial, globus pallidus, lateral, putamen. And then we start to get early signs of these large relay centers, interpretation centers called the thalamus. We saw that on a mid-sagittal slice. They're going to form the lateral wall of early transition of the third ventricle. The third ventricle is always, at least in the healthy brain, is a midline structure. And again, it's going to be found rather adjacent to the thalamic bodies. Here, once again, are certain amounts of choroid plexus. The superior sagittal sinus is going to produce that triangular shaped defect. Basically, what this is venous blood moving directly down the posterior midline of the brain. Continuing down, and again, these slices are very small, very gradual. Things that we know about. Always going to go back to those. Genu, splenium. Oops, sorry about that. Head of the caudate nucleus. Over here are the lentiform nucleus. What I neglected to mention before in my maybe like exuberance explaining the lentiform nucleus is that you have bands of white matter tracts that are also critical from the standpoint of transmitting different impulses to either in and out of the basal ganglia to different portions of the brain. And they form, I'm going to go back a step if I may. Over here, you see somewhat of a V-shaped density right through here, which makes up white matter tracts. And then you see another one on the lateral portion over here, which is another band of white. We break these into what we call capsules. There's one that runs between the head of the caudate nucleus and the lentiform nucleus. This is called the internal capsule. You'll see it bilateral on both sides. Here, outside of the lentiform nucleus, you have another one, band of white, called the external capsule. And although we can't quite see it, there's another small basal ganglia called the claustrum. And what's found between it and the lateral fissure of the brain or the cerebrum is another one called the extreme capsule. So we started off with the centrum semiovalli, had this large burst of white, and now we have interpretation, not interpretations, but rather impulses being carried to different portions of the basal ganglia and onto different portions of the brain. These bands of white are critical. If you were to have a bleed, a subarachnoid hemorrhage, if you were to have trauma, and it was to interrupt one of these capsules, you lose a lot of function in terms of one side of the body versus another. So once again, what you have is caudate nucleus, lentiform nucleus. Between those two, internal capsule. Outside or lateral to lentiform nucleus would be the external capsule. Right over here, in fact, let me go down one more level where we were before. Right over here is what we used to call the Sylvian fissure over here, now called lateral sulcus. You see perhaps better. You see head of the caudate over here, once again adjacent to the anterior horns of the lateral ventricles. You have over here internal capsule, external capsule right over here. We talked about the fifth lobe of the cerebrum. Right over here is where it basically resides. It's right along the portion of the lower portion of the cerebrum, and it stands out as a lobe. You don't see it as a distinct structure. We didn't spend a lot of time on the different lobes because of time, but this area just medial to the lateral sulcus or fissures is where you would find the insula. So when you look at textbooks and they identify the insula, it's going to be right adjacent to this area right through here. Once again, choroid plexus, third ventricle midline structure, always found on either side of the thalamus. A little bit lower perspective, we can see we're starting to leave the area of the cerebrum. Better view of the third ventricle. Posterior horns of the lateral ventricles, they're quite extensive. You have a little spaghetti in here, which is going to represent choroid plexus. And then there's a connection between these two horns called the antrum. And again, we won't spend a lot of time with that. Again, the insula is going to be just adjacent to it. Kind of hard to stand out as an individual landmark. Again, I'm going to continue down just a different perspective. One of the things about the lateral ventricles, you're going to have three major horns or extensions. We already identified for you the anterior horns, posterior horns, and then you have a little area right over here which kind of drop down, which are also extensions of the lateral ventricles called the temporal horns. And they're going to be found in the temporal lobe of the cerebrum. And we're getting a glimpse of them. Oftentimes they're slender, they don't stand out, but they're certainly there from that standpoint. So what we have over here is temporal horns of the lateral ventricles, posterior horns of the lateral ventricles, antrum that connects these two together. Here's the third ventricle, and then you have the thalamic bodies continuing down. We won't get into ultrasound, we get into some of that in my other class. Midbrain, love the midbrain. And we'll kind of look at it from a couple of perspectives. Basically, this is another division as we work our way down the cerebrum that we have a lot of key landmarks. First of all, these ears, I've delved too deeply into those already, I'm sure. These are the cerebral peduncles. They're going to connect the cerebral cortex to the midbrain itself. The midbrain is the head of Mickey right through here. We'll find out that there's some key structures with it, some areas that appear dark, we're going to refer to as the substantia nigra, and there's some other structures in here as well. You have the vermis, which is another connector that connects both hemispheres of the cerebellum. You have a portion of the dura mater that separates the posterior portion of the cerebrum from the cerebellum called the tentorium cerebelli. It has almost that wishbone appearance back over here that indicates the tentorium cerebelli. And then you get into an area, one of the larger cisterns, and we'll get into the arterial circle, which is the new term for circle of Willis. This is called the suprasellar cistern. It's a large collection of CSF. And what makes it really important is this is where you're going to find things like the optic nerves, the optic chiasma. You're going to find in and around it the arterial circle. We'll find that the pituitary gland will also be found in the lower reaches of that area. So this area is one of the important cisterns back over here from that standpoint. The little dot over here is the mouth of Mickey, excuse me, which is going to link the third ventricle to the fourth ventricle. Again, old textbooks refer to it as the aqueduct of Sylvius. In the New Grays, or it's been around for probably 10 years now, excuse me, is the cerebral aqueduct, connects the third to the fourth ventricle. Colleculi, back over here is, I'm going to refer to as the chin of Mickey. And these little two dots right over here. These are paired structures, small in size, very big in terms of their functionality. Basically, you have two pairs up top called the superior colleculi, and there's two below it called the inferior colleculi. What they help us do is in terms of reflex of our eyes, in terms of movement based on auditory stimuli. So when we hear something, what happens is that our eyes are beginning to move before we're ever conscious of it, or before the head moves. Imagine driving down the freeway, you hear a screech behind you, you hear the brakes locking up behind you, and of course you're going to tense up and all that good stuff. But what will happen is that your eyes will already move based on the auditory stimuli of that braking sound toward wherever the sound came from. And in part, it's being controlled or produced by these little colleculi. Really fascinating. As you can tell, I can spend probably a long time just talking about different features, functions, and physiology of the portions of the brain. I really would love to be able to have another opportunity to speak to you about different things about that. All right, circle of Willis is still referred to when I talk to my students when they're going through their angiography, interventional rotations. They still do COW studies. We do a lot of that in MR. In Gray's Anatomy, they've called it now arterial circle, and we'll define each of these over here. What this basically does is that near that suprasellar cistern right over here, you have what we call anastomosis, or connections between various vessels that produce this circle. We'll define each of these for you. But basically, this interconnection of arteries are going to help maintain intracranial pressure and flow of blood to different portions of the brain. We'll talk about what makes it up in regard to this arterial circle. We're going to find the two, actually three major tributaries that form it will be the two internal carotid arteries coming up from the carotid canals or passageways and then entering just adjacent to the cella turcica. And then the vertebral arteries, once it crosses through the foramen magnum, will form a basilar artery. It will also be a major tributary that forms a portion of that arterial circle. We'll see elements of it. Here, from those getting ready for your boards, make sure that you know the 5 components of the arterial circle. What doesn't make it up, rather, is the basilar artery. So what you have is this. We have internal carotid arteries, ICA. They're going to bifurcate. It's going to form one bilateral branch. Oops, sorry about that. Going directly anterior on both sides, called the anterior cerebral arteries, because the space, I abbreviated those ACA. There is an interconnector right through here called the anterior communicating artery. If you looked at a cadaver, you'll see it as quite a small little bridge. We'll show it here on an MRA here in a moment. Extending posterior from the internal carotids, you have the posterior communicating arteries back over here. And then finally, what they connect to is being produced by the basilar artery, the posterior cerebral artery. So again, I apologize for the abbreviations, but keep in mind, you have 5 members, 5 structures that make up the arterial circle. ICA, internal carotid artery, anterior cerebral arteries, posterior cerebral arteries, and then the 2 connectors, the small singular anterior communicating artery and posterior communicating artery. Here's an MRA, somewhat of an axial oblique. What you have over here, kind of not really shown to form, the 2 vertebral arteries coming up. Once it crosses through the foramen magnum, it forms the singular basilar artery. It bifurcates near the area of the suprasellar cistern, near the area of the pituitary gland. It bifurcates off this way and this way, forming the posterior cerebral arteries. Up here, we see the internal carotid arteries coming up, forming the anterior cerebral arteries, the singular anterior communicating artery. What's missing on this one image is the posterior communicating arteries. Again, this is going to produce that circle or that anastomosis of these 5 vessels. So again, it's important for you to know different elements. Let me go back a step. One of the great questions that shows up on these boards is I'll try to trick you and say, does the middle cerebral artery form a portion of the circle of Willis, arterial circle? The answer is no. They go out into the lateral reaches. So the middle cerebral arteries are not part of that circle. If it shows up on your boards, you can write me a note later and say, hey, that's pretty cool. I didn't get tricked. All right, things we've already covered before. I'm not going to delve too deeply or more than that. Cerebral peduncles, midbrain over here. We have the cerebral aqueduct linking the 3rd to the 4th ventricle, vermis, a connector between both hemispheres of the cerebellum. We have the tentorium cerebelli back over here. Oop, let me get my pointer back, which again is that dural cover separating the posterior portion of the cerebrum from the tentorium. A little bit of that straight sinus. We'll see it better on the sagittal over here. would be part of the superior sagittal sinus, and of course optic nerves, which we won't get into from that standpoint. These are kind of the key landmarks that we want to identify this portion. I mentioned before the midbrain, tremendous, a lot of anatomy. We talked about the cerebral peduncles over here. You have red nuclei, which help us in terms of coordination and regulation of motor function of muscle groups. Why they call it the red nucleus is that it has, on a cadaver section, it has a little pink tint to it because of the amount of iron that it contains. We're not sure the importance of that. Over here, when you get into more of function, the substantia nigra has that melanin within it, which gives it that dark tint. And there's special neurons in this area that produces dopamine, a form of dopamine. It's also produced in the GI tract to a certain degree by epithelial cells. And why this is important is that research has been done for many years now, says that certain forms of Parkinson's can occur because when dopamine levels, when these neurons rather begin to degenerate and are no longer producing the levels of dopamine needed by the body will begin to experience spasm of a normal motor function of the large muscle groups. So that's one of the reasons why certain patients with this form of Parkinson's will manifest as some kind of a spasmodic movement of the body. It's because the dopamine that's being produced by these special neurons are being reduced from that standpoint. The tectum, which is the chinamiki, we'll get into colleculi later on, well, I already did, and then the fourth ventricle, which we'll spend a bit more time with that right now. I'm going to skip through this one. The pons is really a cool level. Again, you see some key structures over here. Different pulse sequences back over here is the vermis. The pons itself over here between the what we call the clivus and the pons, either a flow void artifact, we don't see it well on this one, is the basilar artery that always stands out as a key landmark. Back over here, we'll get into a little bit more as it gets lower into what we call the confluence of sinuses. So key landmarks to keep in mind: basilar artery, clivus, pons, fourth ventricle, vermis. Back over here, we're getting into hemispheres of, we're still in the cerebrum, posterior portion of the cerebrum, and fourth ventricle I mentioned before, but also these little arms that extend out this way, perhaps better seen over here, are another set of peduncles that they're called the superior cerebellar peduncles. They connect the midbrain, although it's at the level of the pons, the midbrain to a portion of the cerebellum. Continuing down, you see this a little bit better over here. This is kind of a classic sign. These are temporal lobes of the cerebrum, more of the vermis, pons, basilar artery, clivus, fourth ventricle, superior cerebellar peduncles, vermis over here. And right over here, rather than that nice triangular-shaped defect we had on the posterior midline, it became very confused. This is where all those venous sinuses from different portions of the brain come in, and they converge at this point. And we call that the confluence of sinuses. Typically, you'll see this little defect back over here. It's going to correlate to our little inion or external occipital protuberance. That's kind of where you'd find it. So you see the internal portion of it right over here, that little defect, which serves as a landmark of where you expect to see the confluence of sinuses. The key feature to keep in mind, rather than seeing a nice triangular-shaped defect back over here, it has somewhat of a confused appearance about it. Finally, we get down to the medulla. Here's our vermis. Here's the cerebellar hemispheres. Here's the medulla. These are two little extensions of tissue which are representing the inferior cerebellar peduncles, which connect the medulla to a portion of these hemispheres of the cerebrum. Back over here, you get some flow void art. Go back here a sec. You get into a little flow void artifact over here and over here. Once you leave the confluence of sinuses, it forms what we call the transverse sinuses. Again, this is that venous blood being carried toward the midline of the brain, or actually the midline of the base of the skull. Eventually, what will happen, the blood will go through the jugular foramina, and at that point, once it leaves the skull or the cranium, it forms internal jugular arteries. And then we kind of wrap it up with the medulla. These little guys over here are called cerebellar tonsils. They're really interesting. They are just extension of the cerebellum, which help us with large muscle movement once again, along with some other structures. But there's a condition called type 1 Chiari malformation. And you have your foramen magnum right over here. Here's the two vertebral arteries, which tells us we haven't quite got high enough to form the basal artery. But these little guys here, these little tonsils over here, if they have, because of malformation, extend through the foramen magnum, as the child develops, the brain gets larger, these little tonsils will become impeded, and the child will begin to show loss of motor function. If not corrected, then the patient actually could die from this. This is where we see the Chiari malformation happening. Finally, I want to talk a little bit about the pyramids and olives. Right within this area, we can't see them as landmarks. You have two important areas. The olives are just a motor nuclide, but right along the anterior portion over here, you have two small areas, best seen on a cadaver slice of the brain, little gray structures, and these are called pyramids. And they're going to be responsible for a process called decussation. This is where the nerves are coming up, both motor and sensory, coming up from the spinal cord area. This is where they cross over in this tiny little region right over here through the structures called pyramids. Once again, they are responsible for a process called decussation. Imagine if you have a stroke or injury along this area, along with the medulla controlling certain aspects of cardiac and respiratory function, you can definitely lose motor sensory on both sides if any injury happens to this portion of the brainstem. Finally, I know I've been taking you on a very gradual process. I hope this has been helpful. We have the venous sinuses. This is an MRV, and I gave you more of an oblique perspective. This is going to be found within the falx cerebri, going from anterior to posterior. This is superior sagittal sinus, eventually following the posterior midline. We don't see the inferior sagittal sinus. Eventually it's going to meet up right through here, through the great cerebral vein. There's another vein in this area. This forms the straight sinus, which we'll see above, and then piercing through the tentorium cerebelli as it moves posterior. Where these sinuses collide, we're going to have the confluence of sinuses. Then it forms bilaterally, transverse sinuses, goes through what we call the sigmoid sinus, and eventually it's going to terminate, once it hits the midline, through the jugular foramen, and then eventually form the internal jugular veins. That was a lot of information, I know, with a few stories interwoven.